Hello and good morning. I hope you all are doing well. Thank you to those of you who send me a message to let me know that you're doing well. I really appreciate it. For today's topic, I will start with the regulation of gene expression by non-coding RNAs, the topic that we were discussing last time, and we will focus on piRNAs today. And then I will move on to the next topic, genome editing, with the special emphasis on gene drives. Here are two papers listed. One delves more in detail with CRISPR-Cas9 based editing and targeting the genomes, and the second paper really goes into detail with gene drive systems for targeting female reproduction, especially in malaria vector Anopheles gambiae, the African malaria mosquito. These are very important papers if you want to know more about the mechanisms that we will be talking about today. Okay, so the piRNAs or PV interacting RNAs are the largest class of small non-coding RNA, those that express in animal cells. PV stands for P element induced wimpy testis in Drosophila. So as the name suggests, instead of their expression more in somatic cells, these piRNAs are associated more with the germline tissues. So expression is more in the germline tissues. These piRNAs form RNA protein complexes, just like we talked about siRNA and microRNAs. They make protein complex with Argonauts. Here, the piRNAs, they made protein complexes with PV proteins. These piRNA complex are mostly involved in epigenetic and regulation of other genetic elements in germline cells. So that's the kind of important here, as these are more when we talk about piRNA, we are really talking about the germline cells, the changes in germline cells. So they play a role in germline differentiation. Here you can see that these piRNA, along with the PV protein, because these are piRNAs, PV interacting RNA, they interact with the PV proteins. They can affect the gene expression in two ways, either directly methylating DNA molecule, so that DNA methylation will change the gene expression, or modification of histone proteins or methylation of histone proteins. So either way is directly methylation of DNA or methylation of histones will change the expression of the gene by expressing the chromatin remodeling, by changing the chromatin structure. Okay. So in cytoplasm, these will then lead to translational inhibition or mRNA degradation, more frequently translational inhibition because of the chromatin remodeling. These are involved in gene regulation, transposon repression, genome protection. And these here you can see this is elongation eukaryotic initiation factor. So if it's piRNA binds to initiation factor, then that will lead to inhibition of so that will change the gene expression by inhibiting the expression. Here, this paper on PV interacting RNAs, small RNA with big function, really goes in more details about PV RNAs. So if you're more interested, you might want to read this paper. That's again a really good paper. These piRNAs, as I said, these 21 to 35 nucleotides, a little larger than siRNA and mature microRNA. And these, they can regulate gene expression in multiple ways. Here, just to side-by-side compare both microRNA, siRNA along with the pvRNA or piRNA, you will see that AGO is a protein that's very important for both microRNAs and siRNA, because microRNA and siRNA make complex with this protein, whereas in PV or piRNAs, it's the PV protein that complexes with piRNA. Another really important thing to keep in mind is microRNAs are 20 to 23 base pair, siRNA also in the similar range, about 18 to 21 base pair. piRNAs are a little longer, 25 to 31 base pair. These microRNAs have hairpin structure. Remember that hairpin needs to be first removed in order for microRNAs to be activated or to be a mature microRNA. siRNA are double-stranded RNA that are broken down into small pieces, and these piRNAs are single-stranded RNA. For biogenesis, there are two enzymes that are very important for microRNAs, DROSHA and DICER, because they have to cut on both ends. DICER cuts at one end, DROSHA cuts on the other end of that hairpin loop. For siRNA, DICER is important for chopping this double-stranded RNA into smaller siRNA pieces. PiRNAs are DICER independent, so they do not use a DICER because they're already single-stranded. They're smaller molecules. So here's it shows this is microRNA, comes with AGO, finds the target mRNA, usually leads to the regulation of mRNA stability and translation because it binds on the 3' end of mRNA, usually. siRNAs, they regulate transposon activity. Protection from viral infection is one of the major mechanism of siRNAs, especially in plants. Most of the studies have been with an siRNA as a protection mechanism against viruses, which is also common in animals as well. PV or piRNAs, regulation of transposons is one of the function, and the other functions are still, we're learning about these piRNAs, what their functions are. Because they express in germline, lead to condensation of the chromatin, and then they might have some specific functions in the germline cells. All right, so this is all about non-coding RNAs that we're going to talk about. Next, I'm moving on to the next topic on genome editing, gene drives. Genome editing, because we're talking genome editing, more of the more instead of the classical transgenics, so we have been focusing on more of the tools that are more current tools, for example, CRISPR-Cas9 based genome editing. I'm not going to go to play this video. You can watch it later, but we have talked about CRISPR-Cas9 quite a bit throughout the semester, and probably by now you understand very well the mechanism of CRISPR-Cas9. You understand that this is a very good technique for precision editing of genomes. It has already been employed in medicine, in physiology, in a number of different areas of biological sciences. There are still many other ways where CRISPR-Cas9 can be used, and one of those I will be talking about today is gene drives. So what are gene drives? Gene drives are, there's this video where you can you can watch about gene drives, what gene drives are, but I will go and talk about a little bit more about the different mechanisms of gene drives, what gene drives are, what are the different mechanisms of gene drives. So when we're talking about gene drives, so one of the things I would like to mention is gene drives are a controversial topic, and I will give you the details of what these are, and then you can decide what you think about them or not. The U.S. military, DARPA, the military scientific research branch, is investing a lot in development of gene drives. There are a lot of different projects going on to find a good gene drive that can potentially reshape or override the natural selection. Most of this work, when we talk about gene drives, most of this work has been associated with some sort of control measures for insects, but it's not only insects. It can be used for any animal that has a shorter reproductive cycle, a shorter lifespan as well. For animals with a long gestation period, a long life cycle, for example humans, it's going to be hard to do, gene drive, and we probably don't even need to do gene drives per se, but just keep in mind it's more suitable for individuals with a shorter lifespan and shorter gestation period. There are people who's against it, scientists who are against gene drives. They've been talking about how gene drives could be risky for field trials because we don't know a whole lot about gene drives yet, so we shouldn't be going ahead and using these in the field. But so before going into all this controversy, let's talk about what gene drives are. So gene drives are as I said, these are mechanisms for overriding natural selection. Why do we need to override natural selection? Because when we're talking about genome editing transgenic animals, and if we are going to release transgenic animals in the wild, these transgenic animals usually making anything transgenic, any animal transgenic, place an extra metabolic load on the organism. So that you can say a fitness load. So this individual might be, say it's a plant, so this plant might be insecticide resistant, but at the same time it might have lower yield. So you don't want a plant with lower yield either. So making it transgenic might cause, might improve one part, and the other part it will have a little have a negative impact of that. So these transgenic animals, transgenic plants, they can persist in the wild as long as there is a selective pressure. For example, there is a breeding pressure, there's a pesticide application. So if we are trying to release, say for example, transgenic insects, those transgenic insects will, if these are insecticide resistant, so they will only be applicable when there is an insecticide. So the wild population will be lower at that point. Or these individuals, they can survive when there's pesticide application, because it gives a selective advantage to the carrier. But if selective pressure is not maintained, say there is a disease outbreak like we are seeing now, or there's a war, and then the government is really focusing on that war and not focusing on pesticide spray, etc. That's if that selective pressure is not maintained, then transgenic could be outcompeted by the wild type animals. Over the evolutionary time, those definitely would, but even in a shorter time, these could be overcompeted. So then there comes gene drive. So gene drives are a transgenic construct. These can spread through a population even when their effect on population is negative. So what does that mean is the gene drives, they alter the probability of transmission of an allele to offspring by more than 50% the natural probability. So if you're using gene drives, these gene drives have this mechanism in them that these will propagate themselves into the population. So if you have a few individuals with gene drive, you release those. These will make these other individuals. They mate. All those progeny will have that gene drive as well. So now because you made these individual mated with some of the wild type, and all of that progeny has gene drive, so you have just increased the number of gene drive individuals in the population without releasing those over and over. Now these gene drive containing progeny, that when these mate with wild type population, their progeny will also have the gene drive. So that's the concept, that these transgenic construct, even they might have a negative impact on the population, somewhat negative, but they can still spread through the population. So this technique, gene drives, can be employed for, we can add a gene to gene addition, deletion, disruption, modification, any sort of things that we talked about that we can do with CRISPR, we can do with site-specific mutagenesis, we can do with transgenics through transposable element. All of these, either by transposable elements or by site-specific mutagenesis or by CRISPR-Cas9, any of those, when attached to a gene drive, these can become a new way of controlling a population. Mostly when you talk about gene drives, you will hear about the work on mosquitoes, the mosquitoes that transmit malaria, dengue, or Zika virus, or to control invasive species. So there's a lot of work that has been going on in some of the non-drosophilid fruit fly species that affect, say for example, taffeted fruit flies that affect a lot of different fruit plants, melons and guava and a lot of stone fruits. So all those flies, they can have an enormous economic impact, and to reduce their population, gene drives have been proposed so that we don't have to release the sterile male, for example, over and over for elimination. So that's the concept. There are several labs throughout the world that have been really pioneering these technologies. The one in San Diego, at UC San Diego. Omar Akbari's lab has been on forefront of these technology. Another is at Imperial College in London that has been really doing a lot of work in this area as well. There are other labs, but these are that's kind of on top. Okay, now let's talk about gene drive strategies. How to make these gene drives? How to make something that will not follow the classic Mendelian inheritance of if you have there are two alleles, one coming from a parental chromosome, one from maternal chromosome, or one parent or either of the parent, there's a 50-50 chances of that allele to go into the population, right? So the individual, the offspring can have either, there's 50% chance whether this offspring will get the maternal allele or paternal allele. But when we are talking about gene drive, we are not talking about that. It's skewed a bit. So there are several strategies we will go over today. Under dominance, heritable microorganisms or Wolbachia with cytoplasmic incompatibility, meiotic drives or wide drives, driving endonuclease genes. That's the most important right now is this driving endonuclease or homing genes. That's been one of the main focus area of gene drives, and we will talk about these. And transposable elements. So I'm not going to talk about transposable elements today because we've already talked about transposable elements, and I assume that you already understand that transposable element, whether these are DNA transposon or retrotransposons, they can move from one place to another place in the genome, and by moving from one place to another place, the researchers have used this this natural way of moving into the genome to use these as vehicles for taking the gene of interest to where they want to take it, or disrupt a gene, or add a gene, or replace a gene. So we're not going to talk about transposable elements very much today, but think about it that when you use some of these gene drive mechanisms along with transposable elements, that can be a tool for these modifications as well. Okay, let's talk about underdominance first. So the underdominance naturally occur in populations. So what is underdominance? Genetic underdominance, it's caused by two genotypes that are incompatible with each other. So if there are genotypes within the populations, this one genotype has an allele, other one has another set of alleles. If they mate, all the offsprings will be inviable. So they need to have the same allele, same type of allele. So all of these are inviable. So all heterozygous progeny will die. Here, now this is another example. So these are the allele. Incompatible genotypes will die, which are heterozygotes. So all the heterozygotes will die, but the homozygotes will be viable. So this progeny will only survive if it's homozygous for one of the two genotypes is present. Most of the time, this is the genotype that's more common in the population. So we're talking about the whole population. We're not talking about an individual here. So within the population, there are these multiple genotypes. There are individuals that have these different alleles. These alleles will only be viable when these are homozygous. So the allele, this is the most common allele, usually will be fixed in the population because there are more individuals with this allele. So when these individuals mate, they will be able to produce viable offspring. So these will produce viable offspring, but these only when another individual of this same genotype mates with this individual, then these will be viable. Otherwise, they won't be. So that's how the alleles get fixed in a population, because these are incompatible. Not all alleles are incompatible, of course, in a population, but when these are, that is called underdominance. So that means that although in Mendelian inheritance, there should be, they can be either allele. There will be 50-50% chances of the alleles to show up in a population. But when there is underdominance, it's associated with an allele, then only the individuals that have homozygous allele will be able to survive. Another type of gene drive. So why it's a gene drive mechanism? So this is a natural phenomena. This naturally occur in the population. But if you know what these genes are and you can use these in your CRISPR individuals, so if you use another allele that's not usually present in the population, and so introduce those alleles into the population, then the population, these transgenic individuals will mate with the wild type individuals, and all of the offsprings will be those offsprings will be heterozygotes, and these heterozygotes will then die. So this is population suppression technique. Wolbachia is a heritable microorganism, so that's an example of cytoplasmic incompatibility. So Wolbachia is naturally occurring microorganism in many insects, but it does not find in most populations of Aedes aegypti, the yellow fever or dengue or Zika virus transmitting mosquito. They aren't naturally infected with Wolbachia in the wild. So what happens with Wolbachia is, so if there's a Wolbachia infected individual, and this is an infected individual, so this is female is infected, male is not infected. So this is the maternal transmission of Wolbachia to eggs. Those infected offsprings are fine. If both male and female are infected, then their progeny is fine. However, if there is an uninfected female and an infected male, then this leads to cytoplasmic incompatibility, and this will be sterile. So there will be no offspring. So again, it's a population suppression strategy. As I mentioned earlier, that most Aedes aegypti, or very few percent of Aedes aegypti are infected in the wild. So most Aedes aegypti are not infected with Wolbachia in the wild. So when we release these males, because males don't bite, so it's not risky to release males. Only females bite humans because they need blood for making eggs. So when you release these Wolbachia infected males in the wild, so that these Wolbachia infected males can compete with wild type males to mate with the females, and when they mate with the females, that will lead to no offspring, so that will bring the population down. The early experiments with Wolbachia have been quite successful in Australia, and now Malaysia is trying this strategy as well. So these are just some poster from the Malaysia Environmental Agency or Health Department. What it shows here is that they have this person here, the scientist here, she has these Wolbachia infected males that they're reared in the lab. She's releasing these these males, so expecting that these Wolbachia infected male, these will mate with these uninfected females, leading to cytoplasmic incompatibility, and that will result in no eggs produced by these females. How cytoplasmic incompatibility occurs is not very well understood. The exact mechanism is not known. However, in Drosophila, there's another insect that does harbor this Wolbachia. In Drosophila, the earliest effect of cytoplasmic incompatibility are observed during the sperm chromatin remodeling. So the sperms are not able to remodel their chromatin well, and so these sperms are not fertile, not able to provide the correct DNA to be able to fertilize the egg. This is what the concept is, that wild mosquitoes with no Wolbachia. Here, these are the large population of large numbers of these mosquitoes in the wild, no Wolbachia. As you start releasing Wolbachia mosquitoes, just a few Wolbachia mosquitoes over time, these Wolbachia, you keep releasing over time Wolbachia mosquitoes. So you started it here. These you keep releasing over time, and these will start mating with these females, bringing down the population of so these are not now producing eggs. These females are not producing eggs, so there is no more population of wild type, and eventually these are all mosquitoes have Wolbachia in the population, or the population suppression. So the population is just coming down because the females aren't producing more eggs. Y drive or meiotic drive with a sex ratio distorter is another naturally occurring phenomena in the populations, in organisms, where the Y chromosome, there's these genetic elements, these you can call them selfish genetic elements, similar or sort of selfish genetic elements. These selfish genetic elements, they will propagate into the population even when they have a negative impact. Say, for example, there's a genetic element in Drosophila, in some Drosophila, some individuals in the populations that break down its Y chromosome. So that's bad for the sperms to have that gene, because that sperms will die. But even if it has no positive impact on the organisms, but it still propagate itself, because that's why it's called selfish genetic element. So this selfish genetic element concept comes from the common understanding that the whole genome acts together, right? So the whole genome, there's a lot of different genes, a lot of different elements, they all work together so that the individual is best prepared for any sort of issues. So for example, if there is a repair need to happen, if there is a break, the repair will happen. If there is there's an allele that is harmful, might be replaced eventually by changes. So say, for example, how the resistance happened to insecticides. So these insecticides are killing the organism. So the organism will change its gene in a way that those insecticides aren't acting on that gene anymore. So that's the common theme, is that genome works cooperatively. The whole genome it's a unit that working cooperatively. But then, like any society, any population, there are some individuals that might be selfish. This is not good for them to do something, but they still do it because it's in their own interest. So that's a kind of similar way the selfish genetic elements are as well. So think about transposable element. We will talk about the homing endonucleases later in the lecture. So they are also selfish genetic elements. So these elements, they do not provide any benefit to the genome, but they still propagate themselves. And this is the concept. This is a tool. This is used as a tool here, as driving Y chromosomes to use these meiotic drive, these sex distorters, in a way that can modify Y chromosome to express these nucleases to shred X chromosome. Now, this Y chromosome has this nuclease. This specifically breaks down X chromosome. So now what will happen is when there is a zygote that is destined to be a female, because it has both X and Y chromosome, this Y chromosome will break down X, leading to the population that has Y bearing sperms. So these Y bearing sperms are now more common, are the only sperms, so they will only be making males, not females. So they will it will gradually convert the population, so it's all or almost all males. So this starts out with equal number of male and female, but then there is the sex ratio distorter gene. This enzyme is present on the Y chromosome that will that cleaves or breaks down X. So there will be more of those in the population, then more and more and more, and eventually all of the population is male and no female. So that's another way of using these gene drives for population suppression, population eradication. Okay. Since females are most important individuals because they lay eggs, it's also a common theme in animals is the males, especially in insects, that the males can mate with multiple females, but the females usually mate just once or twice. So that gives having more males their advantage of again mating with the fewer females and then leading to extinction. Driving endonuclease genes, or that are also known as homing endonuclease gene or homing-based drives, CRISPR-based gene drives, or mutagenic chain reaction, all of these are known by all these different names, but these are the same things. Homing endonuclease genes, as I mentioned earlier, just in the last slide, these are also an example of selfish genetic element. These selfish genetic elements, again, they might have a negative implication on the individual, on the population, but they will still propagate themselves. These homing endonuclease genes, these are natural gene drives. They're naturally present in the genomes. These are endonucleases. These are very specific DNA endonucleases. These are encoded either by these are encoded as their own genes within the introns. They are kind of similar to restriction enzyme, but again, they're also different. Their unique target site is much longer than the restriction enzymes. Restriction enzymes, remember, they're 4 to 8 base pair of recognition sequence, depending on what class of enzymes they are. These homing endonucleases, they recognize a target site of 12 to 40 base pair long, so quite long site. It's also their target site is not as specific as restriction endonucleases. In restriction endonucleases, their sequence, their 4, 6, or 8 base pair recognition site must be exactly the same sequence, but that's not the case with homing endonucleases. These they can tolerate a variation in the sequence. Okay. Sorry. Homing endonucleases, these induce cleavage at the target site. These 12 to 40 base pair target site that they recognize, they will induce a cleavage, a break there, and then the cell's natural repair machinery will repair the DNA break by homologous recombination. It will copy the homing endonuclease into that site, in the target site. So now both chromosomes have the homing endonuclease. Now also all of the so that individual is now homozygous for that endonuclease there. So this is called homing. That means that's this homing endonuclease gene has been copied into the broken chromosome. It's called homing. So here This is an example. This is your chromosome with homing endonuclease gene on it. This is the other chromosome that doesn't have the homing endonuclease gene. It recognized the sequence here because it's between these two sequences. So it will recognize that sequence. That's their target sequence. This will be then broken. That will be cut, and this homing endonuclease will copy itself there. So again, this homing endonuclease, this is an endonuclease, DNA endonuclease. It will make a cut there. By homology-directed repair, this sequence will be copied here, and now both chromosomes have this homing endonuclease gene. So these can transmit these horizontally within a host population. And now, if this is, for example, these are sex chromosomes. So these sex chromosomes, when they go the eggs or sperm, they both will have homing endonuclease gene in them. So all their progeny will have at least one copy of homing endonuclease gene, because this homing endonuclease gene can copy itself in the other second chromosome. So this will then copy itself there. So now they are homozygous for this homing endonuclease gene as well. So now this increased allele frequency greater than Mendelian rate, right? In Mendelian, only 50% of offspring should have homing endonuclease gene, but because this endonuclease goes through homing process, then all of the individuals will have homing endonuclease gene. Okay, so this is the concept here. So if we use this homing endonuclease gene as a gene drive, these gene drives will copy themselves onto both chromosomes. So you only have organisms that have one copy. You have organisms with homing endonuclease gene. They will mate. They will make this copy in the other individual as well. So here, so this is the individual that has this homing endonuclease gene drive in it. It mates with this wild type that doesn't have homing endonuclease, because this will have copy in both chromosomes. So let's say these individual made these their offspring will have so some of these will have this copy, because this one has both copies. So both of these coming from one of the parent will have a copy, but these copies will then will duplicate themselves in another chromosome. So all 100% of these will be modified, because these offspring will inherit one copy of each chromosome from each parent, and this parent has is homing endonuclease on both copies of chromosomes. So this will go from one to each of the offspring, and then this will copy again. So their offsprings, all of their offsprings will also have this gene drive, and then their offsprings will also have this gene drive. So again, let's talk about this driving endonuclease strategy, or this homing endonuclease strategy. How we can use this to target disruption of endogenous gene. So this is the disruption of endogenous gene. This is the gene that we want to. This is the homing gene. Here, this is the target gene. This is the target. And again, we just talked about this in the previous slides. This gene can be any gene that's involved, maybe involved in viability, involved in fertility, involved in the parasite transmission. So if you knock it down, that will lead to either non-viable individuals, non-fertile individuals, or individuals that are refractory to the pathogen, such as the malaria parasite or the Zika virus. So if you modify this gene, that will impact the organism in a negative way. So decrease the viability, decrease the fertility, or increase the refractoriness to pathogen. And this, because this will copy in each of the chromosome because of the gene drive. Now all of the organisms in the population have this gene. But a lot of this is a great way, and this has been DARPA has been spending a lot of efforts to develop these homing endonuclease-based gene drives or other gene drives to be able to use these in the field, in the wild, to control the invasive species. Those invasive species can be insects. These invasive species can be other animals. For example, mice has been used. There's a whole different projects going on to control mice population, potentially using gene drives. The caution for this is that because you're putting very high selection pressure on an organism, they will evolve resistance to gene drives. And now, if you think about it, you probably would say that, but these homing endonuclease genes are already present in the population. But think about it: anything that's too detrimental to an organism will not be tolerated in a population. So in a population, if the we do have homing endonuclease genes, all organisms, all archaea, bacteria, eukaryotes, they all have homing endonucleases genes, but these homing endonucleases genes usually don't interfere with the cellular function too much in these organisms where this can survive. If they start interfering too much with the metabolic processes, too much with the viability or fertility, then there's a high probability of getting resistance or acquiring resistance against those homing events. So how individuals can have resistance to these, can adapt or can start resistance, or can initiate the resistance to these nucleases, these homing endonucleases. So one of the examples that was studied in the lab through a directed evolution study, as well, these scientists at Imperial College, they made the lab that's been very actively working on developing these gene drives for mosquitoes. They developed these mosquitoes with homing endonuclease gene drive, and then they followed through multiple generations to see if these after how many generations there is the population, instead of going down, because you would assume that because you're using these homing endonuclease at every population there will be suppression. So since these experiments were carried out in large cages, they knew how many individuals were there, and then after every generation they will count to see it should be a downward slope. It should keep going down, but at what point it starts to either just stay at that point or starts going up. So that will tell that there has been a resistance developed against this gene drive. And with their experiments, they showed that about 8 to 10 generations of the mosquitoes populations can start showing up resistance against the gene drives. 8 to 10 generations, if you think about mosquitoes, is not a long time, because mosquitoes' life cycle is only about 15 to 30 days. So within a year, there's a potential to develop resistance if constantly using, of course, if the mosquitoes are constantly breeding. So these target site variants could be resistant to endonuclease cleavage, and so they are not susceptible to drive. And what does that mean is, remember, for these to work, these endonucleases to work, they need to make a break, right, in the DNA. So that sequence that they look for, the target sequence, that sequence, although doesn't need to be exactly same of 20 base pair, 40 base pair, but this need to be consensus sequence with some nucleotides that will be more that need to be conserved than others. So if the individual can change that nucleotides that is most important for guide RNA to find and bind, or the endonuclease to guide and to come and bind, then that can lead to a resistant mutation. So, for example, this is the example. This is the original target. This makes methionine proline serine, ATGCC, ATCG. Through natural selection, has changed this CCA to CCT, which still code for proline, but the guide RNA, or this homing endonuclease can't cut it here now because of this nucleotide has been changed. So that leads to resistance. So some things to think about gene drives, and these are my opinions. You know, you'll have your own opinion. The idea of spreading gene into a population as a mean of population control is not new. It's quite old. These actually all these different mechanisms of selfish DNA. It was used to call selfish DNA. Now it's more selfish genetic elements. This concept is not new. It was studied in the 1960s, 70s a lot, 70s, 80s. Really saw a lot of work on understanding these different mechanisms. These Y chromosomes, this meiotic impairment, homing endonucleases. These were all studied, or the underdominance. These were all studies back in the 70s. So people already knew about these. There will be already proposed that these homing endonucleases, these underdominance or cytoplasmic incompatibility can be used as a way to control populations. And they were used to some extent. But what's new now is with the CRISPR-based gene editing tools, because it makes the insertion of these genetic elements much easier than the classic transgenic element or transposable element based transgenics. So now it's kind of revived more now that these endonucleases can be tagged with these CRISPR based guide RNA. Wherever we want to make a cut, we can take it there. So if you combine homing endonuclease with a guide RNA, now the guide RNA will, wherever the guide RNA goes, the homing endonuclease will go there as well, and it will copy itself into that portion. Cas9 will make a cut, or the homing endonuclease will make a cut because that's a nuclease itself, and then that will insert this sequence there that is associated with it, or it will delete the sequence that we want to delete. So this makes it much easier to move these elements to a particular place where we want to move it, instead of the homing endonuclease trying to find its own 40 base pair. If that sequence is disrupted by another guide RNA, and guide RNA takes it somewhere else where the gene that we are interested in, then the whole drive will go there, right? So that's why it makes it more compatible to these sort of manipulations. However, one thing to keep in mind is when we're talking about natural populations, these natural populations are much different than the lab populations. The population we keep in the labs are more genetically similar because they come from the same individuals. They have been there for several years. But then the target population, the field population, is they have natural genetic variations. These genetic variations, they include polymorphism in the sequences that are homologous to guide RNAs. And most population in wild will have SNPs, or single nucleotide polymorphisms, that might make these guide RNAs ineffective in the population. In addition to that, there are different organisms, different hosts, different constructs. They might use different rates of non-homologous end joining, or HDR. So that's also varies from organism to organism. So we will have to, if you're using in different organisms, these will need to be tailored according to that organism as well. The current results show that the constructs that we have right now, these are quite far from being able to attain even moderate frequencies in natural populations. So if we release these gene drive insects in the populations, and then there is a experiment now going on in Burkina Faso in Africa. It's just that what we assume in the lab is very different from when you go in the field. So we're not able to get the equal frequency or even moderate frequency of these gene drives working in the population. So just because an element appeared to have a strong selfish transmission advantage doesn't mean that it will spread into the population. However, this is just my opinion, that since we have these technologies, so we need to take these to field in order to improve on these technologies. So unless we know that these are they need more improvement, or they need to be changed in a way that they're more compatible with the natural populations, so we won't know those answers until we actually go take these technologies in the field. So they're both sides of the argument. Okay, and with that, this was about about the gene drives and genetic technologies. For Friday's lecture, I will I will focus half of the lecture on COVID-19 and really more on the virus itself, not the disease, not the pathology, but just going on to more in the virus and viral replication. And then I will talk about, then we will start the last topic, evolution, and I think it's differentiation. And that will be it.